I can't believe it's taken until video 19 to get to putting some data on a 3D surface, which is one of the most frequent ways you see or visualize fMRI data. But there was lots of preparatory work, and now I think we're ready for it, and you will not be disappointed. Let's suppose you're looking at your results, your data, and you put up the contrast for faces minus scrambled. You're pleased with yourself because you've got nice bilateral activation where you are expecting it, where you've hypothesized it to be. You can probably reject your anatomical null hypothesis here. But you also want to look at your other contrast. So this might be a sort of a face area. And if we switch to next contrast, it goes to the next one we specified, which is objects minus scrambled. And let's go to the global max there and overlay that on sections. And well, it's in a similar kind of place to the faces, but we're hoping it's in a similar but slightly different place. What we really want to do is look at both sets together at the same time next to each other so we can see if they overlap or not. It's possible to do fancy things with masking when you go through the results and mask off areas or include areas. We're not interested in that. We just want to see both sets of blobs together. I'm going to show you a couple of ways of doing that. The first is the fancy way to do the 3D render. If you go to render and display, let's just pick up a single participant's render. If you remember, or if you've actually watched the videos where we went through how to extract segment tissue types, extract surfaces, we have got for all the participants this C1 surface.gii file and a render C1.mat file, and those are 3D data for the grey matter in the original space, non-warped, so as it originally was, the subject's actual brain surface-ish, whereas the MW is a warped 3D surface, and the render MWC is the warped and normalized render pictures. So let's first look at, well, why not look at this surface.gii file, and we've done all our analysis in normalized warped template space, so we're going to select that one. It asks us how many sets we want to put on. That's sets of blobs. I just want to put on 2 sets. You've got to keep track of what you're doing because it doesn't necessarily tell you what stage you're at. We're going to select our SPM.mat file, and we're going to do the faces minus scrambled T contrast with no masking, normal correction at the 0.05 level showing everything. And now wants to know what the second set is. Well, it's from the same SPM.mat. We'll just do the next contrast, objects minus scrambled. Again, no masking, normal correction of the 0.05 level with our extent threshold K set to 0 to show everything. Now it asks which colours, RGB or custom. RGB means it will just go through a standard set of colours. First blob set will be red, second blob set green, third blob set blue, etc. Custom, you can specify your own colours. Let's just go for the default RGB. And what it does is produce this 3-dimensional render, and if I move the mouse pointer over it, you can see that it changes to this sort of dotted circular line shape, which means I can just click and drag the 3-dimensional render and we can have a look at where our blobs are. Now here's a problem. If we go back to the glass brain view at the top, we can see our data is sort of on the ventral surface, and our beautiful render in 3D has got a bit of sort of cerebellum in the way. We can't actually see the ventral surface very well, and our data sort of tucked down inside some sulci there, so we don't get a very good view of it from this 3-dimensional render. Even if you use this and look at the effects of interest, it doesn't appear to show up as such large areas of activation as when you look at other visualizations. So if you want to, you can spin this around, look any way you want, and if you want to then use that data in a report when you write it up, you can go to SPM figure and you can save the figure as, and you could save it in any one of a number of formats, or a PDF, or just as an image, say a PNG for example, and then you can just put that into a Word document or whatever you need to use. This isn't a beautiful way of doing it for this data because it's difficult to see what's going on there. So if we go back to the render menu, click on display again, so exactly the same function, except this time it's asking for a render file. Let's go to this participant's structural image, and instead of clicking on the surface.gii file, we're going to click on the render mwc.mat file. And this file just contains a three-dimensional render image, 6 different views. Ask us how many sets we want to put on. Again, we want to put on 2 sets, exactly the same as before. We select the SPM.mat, choose faces minus scrambled with the usual settings, FWE 0.050, and then select it again because it's also got the objects minus scrambled, same settings. The option here, it says brighten blobs, none, slightly, more, or lots. Try the different settings. Typically you'll not want to do none. I'm just going to do lots just so it looks good. Again, the option which colours to use, we'll go for the standard ones. And so here, this is the 6 views. We can't interact with these views, they're fixed images, but we have the canonical views, so we have the occipital view here, and then we have 2 lateral views which are quite useful. We have the faces minus scrambled contrast in red, that's the first one, and then in green we have the objects minus scrambled, and yellow shows up areas of overlap, although they're not necessarily areas of overlap, because this view just shows us all the significant voxels within a few millimeters of the surface. So it's possible for some to be behind the others, and they show up as yellow here. In fact, if we look at this right lateral view, we seem to be seeing the red face area separate from the green objects area, whereas the left lateral view, the picture is less clear, and the view of the ventral surface is obscured by the cerebellum, which is not nice. But again, if you wanted to use some of these figures in a report, you could save the figure as just an image file and then paste that in. You probably wouldn't want to use all these views. A couple of them aren't very interesting at all. So whether you're on a Mac or on a PC, there'll be a tool just for clipping part of the screen. So on the Mac I can use the grab tool, I can go capture selection, and if this is the view I want, I'll just do that, and I've just got a nice little image here, and I can just I could just save that and then put it into another document. One thing you must remember is you can't just paste that into a document and have done. That needs a lot of labeling. You've got to say, it's got to be clear what study it's from. The data represents which participants, one of them, all of them. Which contrasts do these represent? What is the t threshold, or what is the p-value? Is it corrected or not? You've got to explain everything, and it's got to be clear, so someone just glancing at the figure and seeing the legend understands what's going on. Okay, if we look back at the render menu, there is something else apart from the display, which lets us use either this type of fixed render or the interactive three-dimensional render. We've used extract surface before. After we had segmented the structural images, we extracted surface to get these blank renders and 3D surface files. There's a final option called slice overlay, which we'll get to next, but we have to do some preparatory work first. You can probably tell from that these three-dimensional renders aren't necessarily the be-all and end-all, the ultimate way to visualize data. Sometimes they can be incredibly useful, sometimes they can be a little bit misleading, sometimes they're not the best way of visualizing or displaying data, but it's a good idea to know how to do all the different kinds of 3D visualization.